a lot of this we've touched upon already. So we really should be able to kind of go through this chapter really quickly. We should mostly be done with it. Your goal for this chapter is to just really understand what the basic mechanisms are on how the genetics can change for bacteria. So I have brought up numerous times that I think it's pretty amazing that we started off with these anaerobic, single-cell, rod-shaped, thermophilic organisms living, on the earth, and now we have such diversity, and there's no type of sexual reproduction. How did all of that occur? There's limited numbers of ways in which that occurred. The first one would be mutations. So the first part of this chapter is about mutation. Mutations you studied about in your 107 class. So it really isn't really necessary for me to go through and talk all about what mutations are and what are the different types of mutations. But what I want you to really understand is the importance of mutation. Since there is no sexual reproduction, and we only have a few ways of really achieving genetic diversity, mutations then become extremely important in bacteria. So I brought this up before. I brought up the idea of studies that show something called hypermutation, where the organism will start undergoing mutations more frequently. But even if they only undergo mutations in kind of a normal rate, you still end up with something like this, 10 to the 7th to 10 to the 11th. So that's something like 1 in 10 million, to 1 in, what's 11? 10 trillion, something like that. So like on average, a little bit over every million cells, you're going to get some kind of mutation. Whether that mutation is a spontaneous mutation, like it just happens, just like in eukaryotic cells, that during cell division you can end up about 1 in a million, maybe also 1 in 10 million, where there is some kind of mutation. And we think of mutations, I think, more as being something negative, but when you look at the number of organisms in a colony, in a culture, in the natural environment, there's so many that 1 in 10 million, even though it says here mutations are generally rare, it's rare if you're comparing it to the number of organisms, but when you look at the number of total organisms, then it's not so rare. So it does happen. So when you look through these past ones, it just talks about different types of mutations. So I'm not going to specifically be asking you to go through and give me information that's specifically about the mutations, but what I do generally ask is, why is it so important that we understand about mutations? Why is it so important, mutations in genetic diversity of bacteria? And so sometimes I ask that on the exam and I get almost a blank, space there. Like all of a sudden people don't remember why this is important. So you guys all understand why it is so important, because it's one of a few mechanisms in which we can achieve genetic diversity in the bacteria. And it's obviously the bacteria inhabiting every environment is proof of the fact that there is a tremendous amount of genetic diversity in bacteria. This one slide here, I want you to be aware of this as well too. The carcinogenicity testing from the Ames test turns out just to be something that's kind of important for you to hear about, because going into the medical field, you might hear something about, something is a positive Ames test. Even when you're reading about different types of food additives or different things that are being researched by the FDA, and you hear something about a positive Ames test. An Ames test is really a test that shows whether or not something is likely to undergo mutation. it's, if you're testing a particular compound, you're looking to see if that compound can cause a mutation. And they have realized that if something can cause a mutation, there's a good chance it can also cause cancer. So a positive Ames test is something that's like first step at determining whether something is carcinogenic. How come I can't talk today? A carcinogen, but then when I'm saying that, why is it coming out totally wrong? Okay, let me just word it so that I say carcinogen. So there is a correlation there. That's weird. Something's like not connecting. So do you understand about this? So I'm not going to ask you how to do the Ames test or anything, but I do want you to have an understanding that this is something that can be used just to have that first idea that maybe something is going to be a carcinogen. There's a lot of testing in which different compounds have to undergo in order to determine whether it really is a carcinogen. A lot of things are suspected carcinogens. I'm sure if you read about all kinds of food additives and other things, you know, building materials and chemicals that are used in the production, you know, the plastics and all of that. You hear about all of this, and you know, it takes really a lot of research before they finally come out and say something really is a carcinogen. But one of the beginning tests is just making that correlation that yes, this can cause a mutation, so therefore we think a positive Ames test would be an indication of something that is possible carcinogen. So when we go through, then I just want to talk about the horizontal gene transfers. So right now, you're just having an understanding about mutations and the importance of mutations, and then we're going to talk about the different types of horizontal gene transfer. Horizontal gene transfer is the transfer of genes from one mature organism to another. So this isn't like vertical gene transfer, where you have from a parent to, you know, like the progeny that come from this type of sexual reproduction. But with vertical gene transfer, it's just one organism that's already a mature organism transferring genetic material to another. And this can happen in other ways, not even just like a mature organism. It can be an organism that's a dead organism as well. So again, the importance of these are really other ways of contributing to all the type of genetic diversity. And we've touched upon each one of these. But this is just something that when it comes to the test time, people start to get these confused with each other. Part of it is because after this, we're going to talk about a lot of different organisms. So what I'm going to say to you that I really want you to take this to heart is that after this lecture, you need to commit these to memory, because when it comes time to study for the exam, you will have forgotten the difference from these different ones. Okay, just because like it's very subtle, and you're not having to learn a lot of detail about any of these either, but it's still something that can end up being 12 points here or 5 points there on the exam. And if you've forgotten it and now all of a sudden it's like confusing to you, then there goes, you know, 10 points or more. So it'll be fresh in your mind. Make sure you review it. Make sure you commit it to memory, because there's going to be a whole bunch of information, like probably around somewhere around 45 organisms that we're going to talk about, and groups of organisms. So make sure that you kind of commit this to memory. When we talk about the different types of horizontal gene transfer, there's some of this terminology in there, especially like exogenote and endogenote. So if I go to this particular picture, the exogenote, that is genetic material that's being transferred, where the endogenote is the genetic material that's already part of the cell. So you'll see those terminologies going up. So basically this picture, it starts looking a little bit confusing here, but it's really not that bad. Basically, this is what we're looking at as the organism that we're starting with. Just that circular piece that's referring to the nucleoid. So even though in our previous pictures, a nucleoid looked like the little squiggly, dense area in the center, just this is what we're showing as the chromosome. Okay, just that circular piece. And then conjugation, transformation, and transduction are then the three ways. And under each one of these, there are a couple of different ways as well. So that's going to change the cell. And then here they're just showing that there's all different ways in which once you have some genetic material entering the cell, that different things can happen. So the unfortunate part of the textbook right now is that the pictures that go along with each one of these processes start looking confusing because they show you all the different things that could possibly happen. So when we go through, we're going to try to talk about just what happens when it's successful. Okay, so if we have something like transformation, you have a piece of genetic material, it could be just a piece or it could be a plasmid, it's going to enter the cell and somehow it's going to change the cell. It can become part of the chromosome of that bacteria. Okay, but also it could just disintegrate and never come part of it. It could just not have any kind of change at all. So let's just sort of be aware of the fact that just because genetic material does go into the cell doesn't necessarily mean that it's always going to have an impact. That way, when we talk about each one of the processes, we don't have to talk about all the ways in which it doesn't work. Okay, so when you look here, you can see sometimes it just destroys it, sometimes it doesn't self-replicate, so nothing more happens. Sometimes it does, and then it goes on to pass that on to the other organisms when it undergoes binary fission. Sometimes it becomes integrated, but again, sometimes nothing happens and it doesn't work. When it comes to conjugation, you have to have contact with two cells, and they just haphazardly come in contact with each other, and then they haphazardly get knocked apart. So sometimes it doesn't work. So now that we know that sometimes it doesn't work, well, just like every time that it doesn't work, we're just not going to worry about that in the process. Do you guys understand what I'm talking about? No? Maybe. This next few slides are really talking again about things like transposons, where you can have genes that are going from one section of the chromosome to the other. Also, that can happen from a plasmid to the chromosome. And this would be something too that you guys learned about in 107. So I'm going to, again, just kind of stick to what we have to learn about with the horizontal gene transfer. You already know about plasmids. And this is the picture that I really have to give into my other chapter. This is a pili. This is an F pilus or a sex pilus. So you can see how much bigger that is than the fimbriae. So you can see the fimbriae all around that cell. This is such a good picture, and even bigger than the flagella. So in conjugation, what we have is this, where you see here, direct cell-to-cell contact, mediated by the F pilus, and you have then DNA transfer from one organism to another. So when we learn something about the sex pilus or the F pilus, that's what it's involved in. The only problem with this is that the more recent research really indicates that that's not what happens at all. And they're not really 100% sure exactly what the F pilus really does. It may only just connect the two cells, but the idea that the genetic material really goes through the F pilus into the other one seems to have been disproven. I read an article, I don't know, a few months back, where really they were, maybe for the past 10 years, have already thought that this is not what happens. This is kind of what happens like with textbooks, is that it takes a lot to get all the material together and get everything published, so a lot of times you do end up with some information that it takes a while before it gets updated all the way. So this might be something that over the next 10 years, they might be talking about it a little bit different. So like right now, you just need to add into your notes that recent research really is showing that probably this is not exactly how the F pilus does function though. Okay, if I knowingly have information that is really updated from the book, like I feel like definitely you have to be aware of that. I don't want to just like make you memorize information that like they already know is not true. When we talk about conjugation, the simplest form, we talk about something called an F plus cell. An F plus cell is the cell that has the plasmid that is known as a conjugated plasmid or an F plasmid, because it has genes on it that code for the production of that F pilus. Okay, so an F plus cell has a conjugated plasmid. It has a plasmid in it. That plasmid is also known as an F factor or an F particle. And I hate to do that to you too, but it's just that wherever you read, you might hear it referred to in 3 different ways. It could be called an F factor, an F particle, or a conjugated plasmid. Conjugated because it's associated with conjugation. What makes it a conjugated plasmid or an F particle? It has genes on it which code for the production of the F pilus. Okay, it also has genes on it which code for the replication of itself as well. So therefore, if you have an F plus cell, it's going to have a plasmid in it. That plasmid is also called an F particle or F factor that has genes that will code for the replication of itself and the F pilus. So therefore, it's setting that cell up to be able to undergo conjugation. If it doesn't have one of those, then it won't undergo conjugation. It won't form an F pilus. So everybody good there? An F minus cell is just simply one that doesn't have that. At least it doesn't have that same one. It actually could have other plasmids. So if something is an F minus cell, it just means it doesn't have whatever the, is in the F plus cell. It could have other plasmids. Okay, so when you have this where they're saying F plus, F minus mating, that's really just like a way to make it, easier for you to understand what's going on. What's going to happen is that these 2 cells, I know it's hard to see this, when they started adding all these other things that could possibly happen, it just made these slides a lot worse, because basically you start off here and then you're going to follow it along like this. So it's a little bit harder to follow along. But here's your F plus cell, here's the chromosome, just the circle in there. There's going to be some type of chemical reaction that occurs when these 2 cells bump into each other. Because that's basically what happens, is they just arbitrarily run into each other, and then a chemical reaction occurs which starts the whole process. So then you have this, you can see here, sex pilus makes contact with F minus recipient cell. Then you're going to have this cell come closer together. They're just kind of attached, but they already bumped into each other in the first place, so this is kind of misleading looking, this particular picture. But basically what you just need to know is that F plus cell, that particular conjugated plasmid, is going to replicate itself, and then it's going to go into the F minus cell. So this slide before, this is just showing the rolling circle method of how it replicates itself. So even though that seems like kind of something minor, they use this process to help actually analyze the genes on plasmids, because they can stop conjugation at any time and actually see like which genes have made it over. And so a lot is actually really known and understood about plasmids based on this type of conjugation. So that's what this slide is. It's just really showing this rolling circle method. So to make this not confusing means that you have to understand that the plasmid, the conjugated plasmid, the F factor, the F particle are all one and the same thing. Okay, so as you read through it, you see it kind of, referred to in different ways. And that you have an F minus cell, an F plus cell. The F plus cell is going to replicate that genetic material from the conjugated plasmid and transfer it into the F minus cell. So all of what's going on here, this is all just telling like how that's happening, but you can see here that it's happening, and then at the end you end up with 2 F plus cells. Okay, so F plus, F minus, you end up with 2 F plus cells. And yes, it is true that this doesn't always happen 100%. They get knocked apart. Only some of the genetic material can get over there. Even if the genetic material gets over there, it could just be destroyed. It could not have an effect. It might not make any difference. But you don't have to worry about that at this point. At this point, you just want to think what happens with an F plus and F minus conjugation. You have to have a conjugated plasmid, also known as the F factor or F particle, because it is what has the genes to form the F pilus. Right now there's some controversy over what the F pilus actually does, but they do know that it is involved in conjugation, just not in the same way that they initially thought. That if everything is successful, you end up with 2 F plus cells, because that F factor replicated itself and then sent that into the other cell. So now you have the initial cell that had the F factor in it, and now you have a second cell that has that. So is that good? Simple, right? That's pretty simple. Okay, so the only thing is that you do have to understand it, because we go on from here. What happens next is we could end up with something called an HFR cell. In HFR conjugation, you have the F factor, okay? What was the F factor? Also, the conjugated plasmid became integrated into the chromosome of the cell. Okay, so if we go back here, what happened next is that became integrated into the cell. Okay, like we talked about what happens with the viral genetic material when it became integrated into the chromosome of the cell. When that happens, the cell is now called an HFR cell. HFR stands for high frequency of recombination. So you could think of like H high and then FR, either a frequency or a frequency of recombination. Because it's about 1,000 times more likely to undergo conjugation in this form. Okay, so if the conjugated plasmid becomes incorporated into the chromosome of that host cell, it's much more likely to undergo conjugation for some reason. Okay, so the only difference here is that the plasmid is incorporated into the chromosome.
It's the same cell.
Yes, exactly. So if you go to this one right here, you had an F+ cell, and then it became an HFR cell, just simply because that became incorporated into the chromosome. Okay, so any F+ cell can become an HFR cell by just having that incorporated, which is called an episome when it goes in there like that. Okay, so we're good on that?
The HFR cell, was it originally an F+ cell?
Yes. See this right here? You have an F+ cell, and it became an HFR cell. And how did it become an HFR cell? Integration of F factor into the chromosome.
So was it just originally separate?
Pardon?
It was just originally separate and now it's...
Right, it originally looked like this, right, where it was separated, and now it looks like this one. Okay, so any F+ cell can become an HFR cell. How does it do that? It just incorporates itself. So when we talked about the bacterial cell and we talked about plasmids, we said that plasmids could become part of the chromosome. In this particular case, we're just talking about a specific type of plasmid, a conjugated plasmid, but there are other genes on that plasmid besides just the genes that code for the replication of itself and the replication or the production of the F pilus. It could have genes for antibiotic resistance, for production of toxins, and so on. But in order for it to be a conjugated plasmid, it has to have the genes for the production of the F pilus. Okay, so an HFR cell, the donor genes are transferred to the recipient cell, just the same thing. A complete copy of the F factor usually is not transferred. That just means that somewhere it broke up, most often because these take time. When this kind of stuff happens, it could take like hours. So maybe only some of it made it all the way over. But again, that's not that important because we've already discussed the fact that any of these things are not always successful. Okay, so when you look at here, now this is showing an HFR cell times an F- cell. So that's the high frequency of recombination with an F- cell that doesn't have that plasmid or anything. And then it's just showing that you have the transfer over there. And now all these different things here are just showing you different outcomes, which is what I was meaning by the fact that it starts getting more complicated because they just like, instead of making the pictures more simplistic, like this is what happens when it's successful, they're saying this is what happens when only some goes over, this is what happens when it's a longer time. But what you want to think of is that if this was completely successful, you would end up with 2 HFR cells. Okay, so if you had the time, they didn't get broken apart, there was no kind of, event that occurred that stopped this, that you would have an HFR cell times an F- cell and end up with 2 HFR cells. Okay, so if you see this, it's like separate, so that's why it's still called an F- cell, the same here. But you could end up where, like, basically what's happening in all these different ones is you're having, just more genetic material entering the cell. So it starts getting a little bit more confusing than you really want. What you really want to just think about is that if this were completely successful, then you're going to just end up with 2 HFR cells. Okay, so HFR times F minus, you could end up with 2 HFR cells. Okay. Everybody followed that? But you just understand, because of timing, it just doesn't always end up like that. But you could end up with just some genes transferred over. And you could see how that could get complicated, right? Because what if some of the genes that got transferred over weren't the genes that were the ones that had the production of the F pilus? Then you're really not going to end up with a true HFR cell, because it wouldn't be able to go on from there, right? Because in order for it to undergo conjugation, it has to have those genes. Did you just follow what I said, or did that seem confusing? See, it always seems like it's simple, and I don't know whether it's just I do a bad job at explaining this, but it starts like getting confusing as we go. Okay, HFR, the only difference between it and an F plus cell is it's incorporated into the chromosome. When it undergoes conjugation with an F minus cell and everything is completely 100% successful, you end up with 2 HFR cells. The only thing that's making it more confusing is that you have all these different outcomes, but the outcomes just have to do with the amount of time that you have during the conjugation process. If it takes hours, then it's very easy, especially in a lot of environments, for those 2 organisms to not be next to each other anymore. Okay, now, if you have a situation where you have an HFR cell like this, and what made an HFR cell? You have that piece of the plasmid in the chromosome, right? So plasmid could go into the chromosome, it could come back out of the chromosome, right? What if, when it comes out of the chromosome, it accidentally takes with it a piece of the chromosome that it was attached to? Or sometimes it leaves behind a piece of itself. That piece and that cell then is referred to as an F prime cell. So that's now F prime conjugation. Okay, so the result when the F factor incorrectly leaves the host chromosome. What's the F factor? It's just that plasmid, that conjugated plasmid that was incorporated into the chromosome. Now it came back out of the chromosome, but it accidentally left with it a piece of itself, or it accidentally took a piece of the chromosome in which it was attached to. You could see how that could happen. If you go back and you look at this picture, you can see all this is going on here, and when you think about it going back the other direction, where you have this coming back out, it could take that piece with it that it was attached to, or again, leave a piece behind. So some of the F factor is left behind, some host genes have been removed along with some of the F factor. Yes?
Is that kind of considered a mutation?
No. It's just a process that just happened kind of incorrectly. But why is this important? It just seems like now we're just getting like really into the, oh, and then it could accidentally do this and everything. Why is it important? Because we started off with just these single anaerobic, thermophilic, rod-shaped organisms all those billions of years ago, and now we have bacteria in every known environment. How did that happen? These are some of the methods. There's only a handful of methods in which all this diversity was created. Mutations, conjugation, transformation, and transduction. So it starts to become more and more important to notice all the different things that could happen, because these are all the different things that lead to the diversity. Because in this case, it isn't just that conjugative plasmid that went in and came right back out, but now when it came back out, it's actually a little bit different than before. So now if it's transferred to another cell, it's going to be a little bit different. So last time, you had to have an understanding about plasmids and about the fact that plasmids were associated with non-essential properties, right? Non-essential properties. So they were things that could make the cell, survive better. It could be things that make it resistant to antibiotics or chemicals. It could be things that allow it to produce different enzymes, to utilize different nutrients and such. Okay, and the essential properties that are needed by the cell were all maintained by the chromosome of the cell, right, in the nucleoli. So now, if you think about F prime conjugation, where you have a piece of the chromosome also being replicated and taking out, now you have essential genes that are really functional for the cell, not just things that might make it a little bit better or might not have any effect, but now you have some maybe really important genes of the chromosome that now could be transferred to another cell. Do you see the difference? Plasmids are usually non-essential properties, things that might help it, but non-essential properties. What if now you have some of the important properties of the cell that are on the chromosome that come back out attached to it? So now instead of just having a haploid condition, you have a diploid condition possibly, right? Because you could have multiple genes for that particular trait. So that's something that really makes F prime conjugation just that much more different. Because it's not just like, oh, it was here or there. But if you came into this lecture really remembering what a plasmid is and what also the chromosome does, they really code for different things. So in this particular case, you really are giving that future cells additional genetic material and possibly really important genetic material and possibly a diploid condition on the chromosome for those particular traits. So in F prime conjugation, here this first picture is just showing that you had the HFR cell. And how did it become F prime? Deintegration, including part of the bacterial chromosome. So that means when it came out, and you can look at those colors, I know it's kind of hard to see, but you see that there's like blue and red, and I think green on there. So you have, this one's green and red, and this one's like blue and red, and this is red and green, so it's kind of showing you that, it's really hard to see, but it's just showing that you have genes that were left behind and you have genes that it also took from that chromosome. And now it can undergo conjugation with an F minus. And if it does that, then it's going to replicate itself and send that to the other cell so that you would end up in a situation where it's completely successful with 2 F prime cells. This isn't really showing you 2 F prime cells, but if it were successful, you would end up with that. Okay, and the reason why they just have made it more complicated is, like I said, there's a lot of reasons why it isn't necessarily 100% successful. So it's more likely that it's not going to be 100% successful than it is, so therefore a few editions ago, they just complicated all the pictures of conjugation to just make them, more accurate. But it just made it more confusing for the students when you're first trying to learn it. Okay, so F minus and F plus cell. You know what happens, one has just the conjugated plasmid, everything is just straightforward, replicating itself, sending itself over, somehow facilitated by the F pillite. Okay. HFR, that conjugated plasmid becomes incorporated into the chromosome of the cell. If it undergoes conjugation, that's replicated and sent over. That's the HFR. F prime, the HFR came out. It took with it or it left behind some of the genes. And if it undergoes conjugation successfully, 100% successfully, then you would have 2 F prime cells. Okay, just because they have that additional information. So that's conjugation. Conjugation is the transfer of genetic material from one mature bacterium to another mature bacterium, somehow facilitated by an F pilus. Transformation. You guys learned about transformation before. Transformation is going to be so straightforward. And then transduction is going to be easier for you because we introduced transduction already when we talked about the lysogenic and lytic cycles. So transformation, you have the uptake of naked DNA, that's just DNA that is just in the surrounding area from a particular cell. That could be fragments of DNA or it could be plasmids. So it's the uptake of that DNA by a competent cell. Okay? A competent cell just means that it's able to uptake that information or that genetic information. That happens during the log growth phase. So during that time, a particular protein is released that allows the cell to become competent. When you guys did a transformation experiment in 107, even though like nobody in here remembered doing that, but like if somebody remembers, or because I do know they do it, but maybe you guys all missed out on that one. But you add something to those cells to allow them to be competent. So that's something that can be induced just in a setting in a lab. Okay, otherwise it happens naturally. So you have cells that die off, and when they die off, you just have that excess genetic material sitting around. That can be taken up by a cell. It could just be a piece of the genetic material, it could be a plasmid. Okay, that can be made competent artificially, that's what I was just saying, in a lab condition. So the pictures are very easy to follow along. You have DNA fragments or you have a plasmid. All it does is just go into the cell. And of course they show you that sometimes it's not successful, but sometimes it is. And in this case, it came into the cell, and now that cell is different because it has additional genetic material in there. And same thing here, the plasmid went in and now you have that particular cell with the plasmid. So fortunately, that's all you have to know about transformation. Okay, just transformation. You get the uptake of genetic material, and it's just considered naked because it's not part of another cell. With conjugation, that genetic material was inside the cell. It was coming from inside of the cell going to another cell. In this case, that information is just there in the environment. And this is something that happens very, very frequently in any kind of environment because you have a genetic material around, but it's just in that time when the cell is healthy and actively growing in which it would be competent to do that. So any questions about transformation?
That it becomes the plasmid.
No, in here, it just is that it became incorporated into the chromosome. If it was a plasmid and it went into the cell, it stayed a plasmid. However, if this could go on and that plasmid then could be incorporated into that genome also. Okay, anytime you have a plasmid, the plasmid actually can become part of the chromosome. That's called an episome. Okay, in transduction now, you have to think back to the lytic and lysogenic cycle. And so I know that's fresh in your mind because you just had to write that out for the exam. And we talked about the two types already when we talked about viruses. Okay, so we have specialized transduction and generalized transduction. And this basically, at the simplest form, is that you have transfer of bacterial genetic material by means of a virus from bacteriophage. So the transfer of bacterial genes by viruses. Generalized. Okay, so if you look at kind of this original thing, just to remind yourself what happens, right? In the lytic cycle, you have the bacteriophage landing on the cell, and that's this particular cycle. Genetic material goes in, causes the cell to make viral particles. Those viral particles assemble into the viruses and they burst out. And so what we talked about last time when we talked about this is that, well, sometimes accidentally it could take a piece of that genetic material. When it could be just any piece like that, that's generalized transduction. So in generalized transduction, any part of the bacterial genome can be transferred. Because just accidentally during that maturation phase or that assembly phase, it just accidentally takes with it a piece of the genetic material that's in the cell. Where did that genetic material come from? It just was part of the original bacterial chromosome. So this occurs during the lytic cycle. So during the viral assembly, fragments of the host DNA is mistakenly packaged into the phage. So it's just during that assembly process, it accidentally takes with it any old piece that just happens to be in the cell. That's generalized. You think about any piece generalized. So here you see what happens. If you look at the colors, the original chromosome is blue. Here, the phage genetic material is red. So you see like, you know, blue and red in here. And then when it assembled, like 3 of them are showing that they have the original red viral genetic material, where this one has blue because that came from the bacterial chromosome. So what's significant about this is that now this bacteriophage is going to land on another bacterium, inject genetic material into the cell, and that genetic material is now bacterial genetic material, bacterial genes that code for, you know, functioning of a bacterial cell, rather than just genes that would be functioning only to replicate that particular virus. So it's transfer of genetic material from one bacterium to another by way of a virus. In generalized, it's just any old piece like that. And it has to happen during this cycle because it happens when you have the assembly of those particular, you know, viruses, those bacteriophages. Okay, also, when you have the lysogenic cycle, you have, again, the bacteriophage lands on the cell, injects the genetic material in here, but you have incorporation of that genetic material into the chromosome. They were called the prophage. But we found out that it could be induced into the lytic cycle. You could have a temperate virus being induced into a virulent virus. Remember, that could happen with things like UV light or chemicals. Something could happen, or it could just spontaneously happen. So now that you know that it was incorporated, when it deintegrates, when it comes back out, it takes with it by accident a piece of the bacterial chromosome. This is going to be specialized because it takes the piece that it's attached to. So if you look at specialized transduction, this happens when you have had a temperate virus. Okay, a temperate virus is the one that undergoes the lysogenic cycle. So a temperate phage that has established lysogeny. So it's in the lysogenic cycle. What normally would happen is that the bacterial cell would undergo binary fission. It would just continue to replicate the viral genetic material, right? But it could be induced into that lytic cycle. Only a specific portion of the bacterial genome is transferred because it occurs when that prophage is incorrectly excised. So during that deintegration, when it comes back out, in order to go into the lytic cycle, it takes with it a piece of the genetic material that it was attached to. So here you have the prophage. Remember, the prophage is what it was called when the genetic material of the virus was incorporated into the chromosome of the bacterium. It deintegrates, so that's what's happening here, and you end up with this situation here, where some of the bacterial genome is attached to the viral genome. When it undergoes the lytic cycle, that piece of the bacterial genome is replicated right along with the viral genome. So in this particular case, you see all of these bacteriophages that are leaving the cell all have bacterial DNA in them, because when it was induced into the lytic cycle, that piece of the bacterial chromosome is there, so therefore it's telling the cell to replicate the genome, and so it's replicating it over and over. And remember, in generalized, it just accidentally took a piece, so you have just one leaving the cell. In this case, you have numerous pieces of bacterial genetic material leaving the cell because it told the cell to replicate this portion, and that portion already had the bacterial chromosome on there. Okay. Now, again, this just shows all the different things that could happen once it lands on a new cell. But right now, you're just going to keep it more simple and just knowing that all different things could happen, and sometimes it's not going to make any difference at all, but this is something that does happen frequently. And bacteria or viruses transferring bacterial genetic material that has like genes that code for certain toxins, that's something that is like pretty well known with pathogenic bacteria, that there'll be a toxin that is associated with a bacteriophage. So when certain strains are going around, sometimes that's how those strains come to be. Are you guys following this? Because a lot of people aren't taking notes. So either you're like totally understanding what I'm talking about. I mean, I've tried not to make it, too confusing or anything, because I always just think this is kind of easy. This is kind of like straightforward. It's like learning the lytic and lysogenic cycle, because there's so much more that you could have to know. If you read through this chapter, you will see that there's a lot that is known and understood about what happens at every step. You even saw like in just the replication process and where it severs things and where things connect, and how that all, you don't even have to know any of, all you have to know is that it happens. But it is something that when 3 or 4 weeks go by, you won't remember this if you don't commit it to memory like tomorrow. See, now it's fresh in your mind. It's maybe a little bit confusing, but this is how to do it. Make sure before you go to sleep tonight, you understand transformation. Transformation is so easy and so simple. There's nothing to understanding transformation. You've learned about it before. You barely have to know anything about it. All you have to know is that it's a competent cell and it's either a piece of genetic material or a plasmid. That's like it. Okay, then tomorrow, make sure that you commit to memory conjugation. And then the next day, make sure you commit to memory transduction. If you sit down and you try to memorize them all at one time, things start getting jumbled around, like, oh, which thing happened in generalized versus like the F prime particle. But if you just sit and just one night, you just make sure you know conjugation, those 3 steps, it'll be so much more simple like that. So transformation before you go to sleep tonight, conjugation tomorrow, transduction the next day, or vice versa. And then just make sure that you keep it up in your mind, because like always before the test, where you're trying to remember somewhere around 45 different organisms and groups of organisms and characteristics and what diseases they cause and all that kind of stuff, then people are like, wait, what happened during, and you don't want to have to relearn this right before the exam. That's when you want to be memorizing all that new material. So now the fun begins. We have just like really next time a very quick introduction to the rest of chapter 17.